Hello, and welcome to Chapter 1, Part 2 of Genetics, a Conceptual Approach, Introduction to Genetics. So we're going to be looking at Section 1.2. Humans have been using genetics for thousands of years. So a little bit about history. Don't start sleeping on me. History of genetic concepts. First we've got Aristotle in 384 BC. He was pretty progressive for his time because he thought that both male and female contribute to inheritance in the next generation. The Roman society, Romans that lived in that time, 800 AD, so a bit later than Aristotle, were practicing animal and plant breeding techniques. They actually started doing that. In fact, here is a picture not of from Rome, but from the Assyrians, showing artificial selection. So where they were breeding, controlling the breeding of this plant. So artificial selection as opposed to natural selection. Artificial selection is where us humans control what's happening and who's inheriting what. So here's a little timeline here. We've got BC, which is before Christ. I think there's another BC term that I'm not familiar with that takes Christ out of there. But AD, Anno Domini, it's just what happens after we switch over from the time before Christ and the time after. I learned it as after death. But here is where this piece of art, which also tells us some history, this bas-relief, where it hits on the timeline. So it's 883 to 859 BC, right here, where the Assyrians are pollinating. They're doing this artificial pollination of the date palm. So you can see these people wearing some headdresses are actively controlling what pollen comes to this date plant so that they can hopefully get bigger, juicier, and tastier dates from their plant. It's their main form of nutrition. And dates actually have 450 calories. If you go and buy a date and eat it from the store and you eat it, it's 450 calories. So it's a very good source of calories for these people. So they had a vested interest. Anyway, artificial selection started way before our time. We're up here 2100 AD. The wheat that you take for granted these days, that's made into flour and baked into your bread and all sorts of yummy treats, cakes, cookies, whatnot, that is a product of artificial selection or artificial breeding. So we have, compared to the plants in the wild, we have cultivated larger plants that give us larger, more numerous seeds, so more nutrition per plant. They don't scatter before the harvest, so we don't lose that nutrition because of the wind or the rain or the hurricanes, combining both of those things. 3 or more wild species were interbred to create modern wheat. Genetic theories in history, many are disproven. So, we have an idea about something, and then we don't just stop there. We, as scientists, keep asking, is this true? Is this true? Is this, in fact, a fact? Ancient Greeks had come up with an idea of what is called pangenesis, which I'm going to talk to you about in a nearby slide. It's now disproven. Also, they came up with the idea that you could inherit acquired characteristics. I'll also have a slide on that. That has now been disproven. Ancient Romans introduced us to the animal breeding practices, which we still use today. Late 1500s, Robert Hooke, with his microscope, one of the early microscopes, looked and found cells. And so he had an idea that called preformationism. that's now disproven. We're going to talk about that in a coming slide as well. Lastly, blending inheritance was an idea that we came up with, and it's now disproven. So you can see that most of the ideas we had were disproven, and we do know that there's animal breeding practices, those worked out, and thousands of years later, we still use them. So coming back to pangenesis, what is it? Well, and we're going to compare it to what we now support with our data, germplasm theory. So on the left, panel A, we've got pangenesis. I'm just going to go ahead and read these bubbles. According to the pangenesis concept, genetic information from different parts of the body travels to the reproductive organs, where it is transferred to the gametes, sperm and egg. Sperm and egg come together, and then you get a zygote, and every part of the body contributed to the next generation. In contrast, we've got the germplasm theory. Again, I'm going to read these bubbles. According to the germplasm theory, germline tissue in the reproductive organs contains a complete set of genetic information that is transferred directly to the gametes. So it's only coming from the germ cells, the germ line, and not the whole body, not the somatic tissue, only the germ tissue where your organs are located. And so sperm and egg are created through the process of meiosis, and those come together in fertilization to produce this zygote, which is going to be the next generation. So we know that the theory on the concept on the left did not become a theory. It's incorrect. But the concept on the right, germplasm theory, is correct. And that's because we've tested it multiple times, multiple scientists over many, many, many years, and it still holds true. Here's another incorrect hypothesis or concept. This idea of inherited acquired characteristics the Greeks came up with. They said, hey, the giraffes, for example, got their long necks because generation after generation they had to stretch up and eat those leaves, and they passed their stretched neck characteristic down to the next generation. This was before we knew about genes and so forth and how those work. That turned out to be wrong. For example, you can't go to the gym, build up your muscles, and pass that down to your baby girl or baby boy. Even though it was wrong, we held on to that incorrect belief for a long time. Other incorrect hypotheses: preformationism with Robert Hooke. In his microscope, he was looking at cells, and he came up with this idea of the homunculus. A homunculus is shown here on the right, illustrated inside a sperm cell, as a teeny tiny human that was completely formed with all of its organs, and much like one of those sea monkeys that you put in your water and it inflates into a big thing. That's what Robert Hooke imagined, is that everything was there, is fully developed, it just had to grow bigger. And that's what preformationism is about. We're preformed, all we have to do is grow bigger. There were people who believed that this homunculus existed in a sperm, and others who believed that the homunculus existed in the egg. So we have the oocyte. That's another term for egg. Ovist versus spermist. It's incorrect. Blending inheritance is the idea that you mix your genes together, very much like paint shown here on this palette over here on the right. And once they're mixed, you cannot separate the genes in future generations. Mendel definitely disproved that. When we look again at inheritance of traits, we're going to see that in some generations, you don't see the recessive phenotype, and but you can see it appear in subsequent generations. So, in fact, the genes can separate in future generations. So both of these concepts are incorrect. So make sure you study this table for your upcoming exam number 1, table 1.1, early concepts of heredity, which ones are correct and which ones are incorrect, so that you may score awesomely on your first exam. Other modern geneticists to know: August Weismann, germplasm theory. Little story about him is that he's like, okay, if pangenesis is correct and you have to have all parts of your body into your sperm, or all parts of your body are represented in your egg, then if I cut off the tail of mice and, I cut them off and then I let those mice breed, what would you predict would happen in the next generation? Well, if you're somebody who is a pangenesis advocate, you're going to say those mice are going to be born without tails because they don't have the genes from those tails because we cut those off. Or we didn't know about genes then, but we don't have the characteristic of a tail because we cut the tail off. Of course, I bet you can guess what happened. If you, the baby mice did have tails, and so that disproved pangenesis and supported the idea of germplasm theory, that you only need to have those traits inherited by your germ cells. Thomas Hunt Morgan is another need-to-know geneticist. Everything in yellow you need to know on this slide. Yeah, there's a lot of yellow. 1910, he discovered the first mutant in fruit flies. And he supported the idea with his research of transmission genetics. Remember, we studied that it's where genes are passed down from one generation to the next, and you can trace that with the pedigree chart. So this was back in 1910, so over 100 years ago now. Fisher, Holden, and Wright, they came up with the idea of population genetics in the 1930s. You don't need to know 1930s. I should, however, have highlighted Mendel and evolution theory, and I will on the PowerPoint that I post in yellow. You should know that that's a combination of Mendel's work and the evolution theory. Yes, Charles Darwin. Okay, so Fisher, Holden, and Wright, population genetics. How gene frequencies change in a population over time. How gene and allele frequencies change in a population over time. Population genetics, Fisher, Holden, and Wright. Watson, Crick, Wilkins, and Franklin, you should be familiar with most of those names. In 1953, came up with DNA structure. We're going to talk about that in a later chapter. Gilbert and Sanger came up in 1977 with DNA sequencing technique. They are chemists. This is a chemical technique. This is telling us the order of the nucleotides, C, A, T, G, and so forth, in a piece of DNA. Carrie Mullis from North Carolina and Michael Smith, not from North Carolina, in 1980s came up with the polymerase chain reaction, or PCR, in which we can amplify a teeny tiny piece of DNA into a lot of DNA that we can now study. Watson and Collins, yes, this is the same Watson from up here with Watson and Crick. Watson and Collins and a company named Solaris collaborated in 2003 to publish what we know as the Human Genome Project, and they also published a lot of model organisms because it's not ethical to experiment on humans, but we can experiment on model organisms, and those model organisms share a lot of genes and cell structure and behaviors and the whole central dogma of molecular biology in common with us humans. And so we can learn a lot from model organisms. The Human Genome Project, as I said, was a project in which we used this Gilbert and Sanger technique to actually find the order or sequence of the nucleotides in a piece of DNA, as shown here by this graph. That we do that with fluorescent dyes now instead of radioactivity. And so each letter A, C, T, and G has a different fluorescent dye attached to it. And so if there's a T in a position, you're going to get a red peak. If there's an A in a position, you're going to get a green peak, and so forth. And so we can find the order or sequence of the nucleotides in a piece of DNA using this technique by Gilbert and Sanger. And the goal was, of course, to sequence the entire DNA content of a human cell, which we did for the most part. There's some areas that are harder to get the sequence of because of the way they are structured. And anyway, we've got most of it. And so we also have sequenced a lot of model organisms. And I'll go ahead and answer why. It's because they have genes and cellular processes in common with us. And so we can find out a lot about ourselves by studying model organisms. Future of genetics includes having contributions to DNA databases to help cure human diseases. So this is a term that you will want to know going forward. SNPs, or single nucleotide polymorphisms. We've got databases with them. What is a SNP? It's illustrated here. It's just as the name suggests. One single nucleotide is different. It's polymorphic. It's different between individuals. So we have a purple, a blue, and an orange individual here. And they have sequence that's identical except for this one position, the SNP position. The purple person has an A in that position, the blue person has a G in that position, and the orange person has a T in that position. So this is a difference between these individuals, a single nucleotide polymorphism. If you get a whole bunch of these, let's get 20 single nucleotide polymorphisms from different places in the human genome, that's like having a DNA fingerprint. And this is something that we can use to track criminal activity, for example, or who's your daddy, paternity tests, and so forth. So your identity is in your DNA as well as in your fingertips. Okay, so we study these small pieces of DNA throughout the genome and how inheritance of the SNPs can be correlated to human disease or traits. Example, myocardial infarction. Are you more likely to have a heart attack based on your SNP pattern or not? Are you more likely to have a certain eye color based on your SNP pattern or not? Okay, so disease also. Other traits, yes. Bioinformatics is the combination of taking these databases that we've created with human sequences and model organism sequences. Oh, I gotta zoom out just a touch. And combining it with molecular biology. Okay, so we got that molecular biology, and we get these databases in the computer. We combine it with our knowledge of molecular biology, how things work in a cell, and we can compare the genome, the transcriptome, that's all the RNA in a cell. So the genome's all the DNA in a cell, transcriptome's all the RNA in a cell, and the proteome is all the protein in a given cell. We can compare these between species such as humans and gorillas and these other primates using these databases. So DNA, RNA, protein sequence comparisons, ultimately we can figure out what the differences are between individuals in different species and individuals in the same species, including us and our SNP patterns. This is part of what is needed for population genetics and for evolutionary biology, which, population genetics is basically a subset of that. So this leads us to being able to develop designer drugs. What do I mean by designer drugs? Well, I'm going to get to that. Sequence, if we get the sequence, if you go to the doctor's office and they take your DNA, and you guys know, you've done 23andMe perhaps, you sequence all your DNA, that's your genome. You can, hopefully, they're working on being able to do this in a doctor's office using a little like thumb drive looking thing that's called Minion. There is a company that had been working on it, Oxford Nanopore Technologies in the UK, where you can buy this thing and sequence your genome for $1,000. And here's what it looks like. Looks like something that can just a little bit bigger than a thumb drive that can plug into your USB port of your computer. You put your DNA in there, it sequences it for you, and then hey, can I take this drug? Will it complement my genotype, or will I have an adverse reaction to that drug? And that's what we mean by designer drugs, designing it so that it works the best with your genotype and your phenotype, what your DNA is and what your traits are. So here's an exciting model with her designer DNA on. Other health applications. Well, if you've got your PowerPoint open and you click here, there's a little article for you to read from The Atlantic, where in the past we have used these little mini ions to help ID infectious viral strains of Ebola in Guinea back when that was a big epidemic. They identified 142 different genomes for this one virus in 6 months using this technology. They also used it to look at Brazilian Zika strains, that different virus that caused birth defects in human babies where they had really small brains and really small heads. They can also identify frogs, plants, etc. And I bet you there's somebody out there using this for COVID-19. There are more than one strain of COVID-19, and it can mutate quite rapidly, especially with its incredible replication and transmission rate. So maybe someday folks are thinking they can use it for something like the Mars mission. I'm just happy if they use it for COVID right now. But Mars mission, if we really can't stop polluting our planet, we may have to go find another one. And it might be good to find out if life can be sustained on a different planet. I'd rather take care of this one personally. So fundamental concepts in genetics, eukaryotic versus prokaryotic. Genes are the fundamental unit of heredity. Genes come in multiple forms called alleles or copies. Copies of genes are called alleles. Genes confer what we call phenotype. That's your traits, how you look, how you act, how you digest, how you metabolize. Genetic information is carried in DNA and it can be carried in RNA. For example, COVID-19 is an RNA virus. It's a single-stranded RNA virus. HIV is an RNA virus. Zika, was it Zika? Oh, Ebola. One of those two is an RNA virus. I forget. Maybe both of them. I'll have to look that up. Maybe you can look it up if you're really curious. Genes are located on chromosomes, and chromosomes separate through mitosis and meiosis. DNA is transcribed into RNA, is translated into protein. Mutations can cause permanent changes. Some changes in your DNA can be permanent. Some traits are affected by multiple factors, not just one gene, but multiple factors, including the environment. Evolution is genetic change, and the genetic change occurs first. That's what you have to remember about evolution. The change in the DNA is already there. And then if there's a change in the environment that favors that genetic change, it will outcompete other organisms. So those are some concepts you should know. If you don't, please refresh your memory in chapter 1 of your textbook. 3 main types of genetic study that we looked at are molecular, transmission, and population. Genetics is important to our lives. We talked about the Green Revolution, where we fed more people because of our genetic modifications that we made to plants. We know that disease is often caused by differences in our DNA. For example, recent reports indicate that COVID-19, you're more likely to be susceptible to it if you have blood type A as opposed to blood type O. You have the least susceptibility with blood type O. We do not know if that is specifically because of your blood type or because of a gene that is near the genes that code for your blood type. So, but it's still an interesting point, and I'm blood type O, and I'm still going to be wearing a mask and washing my hands, etc. Thousands of years it took, especially the last 100 years, for us to understand heredity, and there are still questions we don't know the answers to. And that's why we still have science and research. Now, can we use our genetic knowledge to design personalized drugs? That would be the next frontier. Make sure, and I'm not going to have this for every single PowerPoint slide, but it's a given that your assignment is always to study the actual chapter material. Believe it or not, Dr. Silverkey was an undergraduate at one point, and I did take notes in class. I did attend every class that I could. I did read my book. I would do it out loud sometimes because some things didn't make sense to me unless I read them out loud and had my ears hear my own voice say it. So please make sure you're studying. I mean, I also wrote in my textbook, which my daughter thinks is an abomination, but whatever you need to do to study and get that information in your head, do it. Sapling Online, you're going to need to create an account to do that. There are required activities in there. Begin doing the available Sapling Plus assignments. The animation activities are graded. They are required. So you watch an animation, you study that animation. I recommend that you watch and study it more than one time before you take the quiz because they are graded. Reading quizzes, that's extra credit towards your overall exam score, but you have to do it before each exam occurs, and you have to score 70% or higher. So again, you need to study before even doing the extra credit, please. All right, that's it for this one. Talk to you next time.